We're going to talk about cell culturing, which is a really important method to creating large quantities of particular cells in a short period of time for an intended use or benefit to us as humans. So we're talking about cell culturing. There's sort of three main steps. These may vary from species to species, animal to plant, but there's sort of three overarching steps to the process. So step one, what we're going to do is we're going to take the required tissue from our original location, and this required tissue is going to be dissected to expose the cells, the particular cells which we are culturing and we want to rapidly create more of and create a larger sample of. Second step, we're going to need to add some protein digesting enzymes and this is to release cells from connective tissue. So when we're taking a sample of cells, we're not necessarily going to pull straight out the desired cells. There's going to be some connective tissue and other sort of tissues surrounding it, so we can use these protein digesting enzymes to try and break some of that down and isolate the particular cells that we're after. And then our third step, we're going to place our cells in a culture medium. So this is going to be a liquid containing a number of different things in order to create optimum conditions for these particular cells to be able to grow and thrive. So this may contain, depending on the type of cells, a number of different things, including salts, sugars, growth factors to stimulate mitosis, and possibly antibiotics. This is going to be in order to try and curb bacteria growth. Now, if we're talking about humans or human cells, these are going to be stored at 37 degrees Celsius for humans, because this is human body temperature. So if we're dealing with human cells are going to thrive at 37 degrees Celsius. Now, cell culturing is really useful and beneficial until we reach something called the Hayflick limit. Now, the Hayflick limit, we're talking about 30 to 40 generations, so 30 to 40 divisions of cells, and we start to see a slowing and then eventually a halting of division. So there's kind of a limit or a maximum amount in which in a culture medium cells are going to keep reproducing and recreating until we release this Hayflick limit, where the culture cells aren't going to be dividing and creating any more cells. So cell culturing is good to a certain extent, but then we obviously reach this kind of limit we call the Hayflick limit. So why are we interested in culturing cells? So why do we culture cells? Well, there's a number of reasons. Some you may have heard of, some may be more familiar than others. One of the most interesting ones is cloning. So we're talking about cases such as, I'll put in brackets here, Dolly the sheep, which you may or may not have heard of. So a sheep called Dolly was actually cloned and required sort of cell culturing techniques in order to make this happen. But this is a pretty rare one. We don't, as humans, I guess, clone too many organisms. More common one would be medical testing. So we can use culture medium to test things test medicines rather than something like an animal, for example. We don't have to deal with that whole ethical issue. Cell cultures have a large role in the creation of vaccines, and also producing things like insulin. So beneficial or important medicines that occur naturally in the body, or things that people need that occur naturally in the body, we can use cell culturing to try and create more of something like insulin in order to distribute it to people that require it, that might not be producing it themselves as they should be naturally. So there you have the 3 main steps to cell culturing and some of the important applications that we see for humans.